Welcome everyone. Please take a moment to congratulate your children. They've done a wonderful job, doing research and writing the research paper. So today is their final thesis defense, where they're going to present what they did research on. And so this is the thesis defense and oral presentation. This is what typically happens in any PhD or any research that people do. And I wanted to recreate that for our students. Okay, so this is me, Joe. I'm a success coach, academic study skills coach, and a college counselor. And my purpose is to help guide kids to reach their maximum potential. Okay, so these are some of my certification. I'm a Tony Robbins certified life coach. I'm also a meditation instructor. Sometimes it doesn't feel like that. And I'm a college counselor from UC Berkeley. And this is who is on my team. I have Brooke Schaffner, who's a wonderful essay coach. I have Clark and Jeannie. They are ex-admission officers. Jeannie is ex-Columbia admission officers, and we work together. Then, of course, we have Dr. Raj, with whom we did the research. Prior to that, I had Dr. Ganapati. I have Neetu Rishi, who's a Dale Carnegie trained instructor, and Ram Prasad, who is an AI machine language expert. Okay, so basically, what did your kids do? Students do? They did research, they picked their own topic, and they worked with Dr. Raj and myself. Dr. Raj was pretty much for the technicality and going deeper into the research. With me, they learned how to structure, they learned scientific inquiry, they learned data, statistics, all about the nitty-gritties of the research. And of course, a research paper is valid only if it's peer-reviewed by an expert, and that's why we have Dr. Raj. And the students are lucky enough to get an opportunity to sort of submit their papers in the Journal of Scientific Research. My younger son also did research during spring. He's not yet, so I'm just waiting for a couple of days so that he can submit his. What were the research results? The students developed a deeper understanding of the subject matter. They picked their own topic. They drove everything. They were fairly independent. And then they wrote a 2,000-word research review that was peer-reviewed by Dr. Raj. And now they're going to present their final thesis. And started, the first person to present is Sarthak Mohapatra. You can go ahead, start sharing your screen. I've given you permission to share. So everybody, this is Sarthak. He's an 11th grader in Robbinsville. His topic is Parkinson's disease progress. Current G37. Excuse me, could everyone please mute yourself, please?
So as she said, my paper is about Parkinson's disease. And I did my paper about the biomarkers, the ones that are currently used, and GPR37. So what my paper is about is, just as I said, it's about Parkinson's disease and its biomarkers. And one of the research questions that I had to ask myself during my paper was, why haven't we found a way to beat Parkinson's yet? It's a disease that's been talked about for hundreds of years, so you'd think that we'd have it figured out by now. But the problem is the biomarkers that we use. They're not really effective. So it's not like scientists have just been sitting around and doing nothing, right? And throughout all of the research papers that I looked at during my time writing my paper, I noticed a few key things. These researchers were doing detailed analysis of the clinical features of PD, such as the motor symptoms and the non-motor symptoms, and they were compiling lists of numerous biomarkers for the diagnosis of PD. But for right now, we don't have biomarkers that are 100% reliable or effective. And the main focus of my paper was about new promising biomarkers, such as the GPR37 receiver found in the cerebrospinal fluid. And this is what I was looking for during my time. So it seemed really promising, and I decided to do more research into it. And I found that it was really great for experimenting and implementation due to its location. And also, I found that this receiver is truly extraordinary because the two most common neurodegenerative diseases in the US is Alzheimer's disease and Parkinson's disease, and they're extremely similar. So biomarkers that diagnose one usually tend to make mistakes and cause false alarms, diagnosing both diseases. But this one did not do that, and it only showed spikes in Parkinson's patients. So it was pretty easy for me to conclude that due to its success in its first clinical trials, the receiver can be considered a pretty useful biomarker, and it had quite high efficacy. So going back to my original point as to why we haven't solved Parkinson's yet, I mean, it's tricky, right? With its closeness to Alzheimer's, finding biomarkers that are 100% reliable and have high efficacy are really difficult. But with the discovery of the GPR37 receiver, the future of Parkinson's diagnosis looks promising. It's basically a miracle chemical. There's no false alarms, and it's really accessible. But, of course, this means that we need more testing. Only a couple trials have been done, and in order for us to prove reliability and efficacy to a point where this can be used throughout the world and finally solve Parkinson's, we need more research. But yeah, that finishes my presentation. Thank you.
A round of applause to Sarthak, and please questions in the chat. Next, we have Arshi Park. She's 11th grader from Plano, Texas. Her topic is the effect of COVID-19 on human body's nervous system. Arshi, please go ahead.
So can you see it now?
We can, yeah.
Okay, so this is my just title page, and this is a picture of me from last year. And so, hold on. This is going to be the structure of my presentation today and what I'm going to cover. So the first topic area that I investigated was neuroscience, which is the study of the nervous system, which includes the brain, spinal cord, the peripheral nervous system, and neurons, and the study of all of its functions. Another topic area would be epidemiology, which is, in short term, the study of diseases. The last topic area I covered would be anatomy, which is a branch of biology concerned with the structure and function of humans and other living organisms. A specific research question that I set out to answer was, why does COVID-19 have such a big impact on our body? But I wanted to go more into a specific area of the body and chose the central nervous system because its complexity interested me. The objective of my research was to find out why and how the nervous system is being affected by SARS-CoV-2, or more commonly known as COVID-19. Some current research that I have actually included in my research paper talks about how experts at Johns Hopkins University have done a recent study summarizing that there are 4 ways that COVID-19 can affect the brain. They also state that they haven't finished their research yet and expect to find more ways the brain is affected. The results of the analysis that I have done on the data allow me to believe that COVID-19 not only enters through the nasal cavities in the nose, but also retrograde transports and connections in the brain. But the most significant findings from my data is knowing that COVID-19 fabricates something that does more harm than good in the inflammatory response that could cause the majority of the organ and tissue damage seen in this virus. Oh, I think I deleted my conclusion on accident. But, and for my conclusion, I wanted to restate my original question, which was, why does COVID-19 have such a big impact on our body? But of course, once I put more research and detail into it, I came up with the question and the topic of my research paper. What effect does COVID-19 have on the human body's nervous system? Some contributions I've made is that I've compared the damage of the nervous system from COVID-19 to outbreaks in the past, including the Spanish flu and Ebola. Thank you.
Yeah. So everybody, round of applause. And guys, you need to ask questions because you will be judged based on your questions. You have to ask 2 questions. So every presenter needs to have asked a question. Got it? Next, we have Laiba. Laiba is the 10th grader from Monroe Township. Her topic is cell metabolism in neuroblastoma. Okay.
Laiba Majid. I'm from Monroe Township. I go to Monroe Township High School in New Jersey, and my thesis defense is going to be about cell metabolism neuroblastoma, and this is a picture of me. Okay, so for my introduction, I'd like to start off by stating my topic, which is cell metabolism neuroblastoma. But more importantly, my research questions were, how does cell metabolism occur in the neuroblastoma? And then more importantly, going into depth about the difference between how this is between the normal, average, ordinary human versus the people who have this certain disease, as the brain is part of the central nervous system, which is responsible for carrying out most of all the signals in the body. So if something occurs in this part of the brain affecting the energy or signaling, it should have a big toll on the body. And that's what made me interested in this topic. For my objectives, I would like to differentiate how cellular metabolic process works and explain like why the processes occur in this certain way, and like kind of go into depth about why, because the whole question for me is always why, and I'm super curious about these types of things. This is an image about where neuroblastomas can occur. They usually occur in infants or in the embryo, which is why it's kind of easier to notice, especially during X-rays and fetal scans. And usually these tumors can be operated on by fetal surgeons, and these are just main places. For my literature review, a relevant research topic that I was actually using for my STEM Academy thing is neuroplasticity in infants. And for those of you who don't know, neuroplasticity is the ability of the brain forming, which is basically how the signals and connections of the brain really form and work. And the connection between these two is, for the developing brain, they need energy for the neurons to be able to send signals. So if there's any problem in the metabolic processes, they won't be able to send signals to other parts of the body, which could really impact the body in a negative way and help the infant not be able to grow up right. This is just a little GIF of how neuroplasticity is in simpler terms. Methodology and results. I found the data and results in 3 main steps. So I collected the data, and then I did data analysis, which is part of results, and then I found the significance of these findings. So for data collection, I basically collected an average of a sample group and how much ATP is produced per person. I found the average number of that, and I compared the number of moles of ATP produced per glucose molecule. For my results, I found that the Warburg effect, or for those of you who don't know, the way that cancer neuroblastomas undergo cell metabolism, they had the most median amount of ATP per glucose. This is a combination of aerobic and anaerobic methods for healthy respiration. For the significance of this finding, I found that this phenomenon was more efficient and produced the ATP faster. This basically means that throughout this process, although there's a median amount of glucose, because of the cell checkpoints, it went by more quicker, which allowed the cells to replicate and multiply quicker, which is one of the reasons why cancer cells are very rapidly increasing. And these are just pictures of the Warburg effect compared to the normal cell. For my conclusion, my research question again: how and why does cell metabolism occur? And this is a picture that I used in my paper actually, and it shows the difference in differentiated tissue versus the proliferated tissue. This basically shows the average amount created, and this is through the Warburg effect, and this is through the regular ordinary human. This is also another table that I put in my paper. It's basically comparing the amount of moles of ATP produced per glucose. You guys can take a look at that if you want. And contribution, I aided in research towards the particular brain cancer neuroblastoma, talked about cell metabolic reactions occurring in every cell and how these processes are greatly varied per malignancy. And that is all.
Bravo. Beautiful presentation. Very, very colorful. Everything, lot of GIFs and everything. Beautiful, Laiba. And as you can see, guys, I mean, who knew a high schooler could talk about oncology or neuroblastoma? That is the level of detail that these kids have displayed. I mean, there's so much that I learned from them. Got it? Okay, so next up we have Abraham. Abraham is an 11th grader from Michigan. His topic is action mechanisms of complementary and alternative medicine therapies for rheumatoid arthritis. Okay, so guys, if you finished your presentation, please check the chat for questions.
Okay, so hi everyone. My name's Abraham. I'm from Bloomfield Hills High School, and my paper looked into action mechanisms of complementary and alternative medicine therapies for rheumatoid arthritis. So rheumatoid arthritis is a disease that affects millions. It causes discomfort and pain within joints. current issues with RA treatment, which is rheumatoid arthritis abbreviated, is the fact that they have extremely low efficacy rates and increased chances of infection within joints. Current methods are called NSAIDs and DMARDs. So I looked into alternatives for RA treatment, one of which was botanical treatment. Botanical treatment consists of natural substances such as herbs and oils. And another one I looked into was anakinra treatment, which looked into blocking the inflammation protein of interleukin-1 within a joint. So testing was promising. A lot of testing was done on this. However, they didn't show great efficacy rates, and therefore I wouldn't consider them great over the normal methods. So how I analyzed this data was I assessed each treatment based on their efficacy and their toxic profile. Their toxic profile being the rate at which they could cause infection within a joint, which is an issue with current methods. Botanical treatments did solve the issue of infection within the joint, as they are natural substances and therefore couldn't really cause infection within the joint. And anakinra treatment did run human trials, unlike many of the botanical treatments. However, they showed low efficacy and really couldn't be considered unless normal methods didn't work. So in conclusion, my paper, again, looked into alternative treatments for rheumatoid arthritis and took this data and collected it into one place. It did solve the issue of infection and toxicity within the joint. However, efficacy is still to be solved due to low testing. And yeah. Thank you.
Yeah, very good, Abraham. Very, very, very nice. And guys, the reason why I wanted you to present is you're going to take the same thing and go to school and do it with your teachers, with your counselors and everything. It's going to directly reflect in your recommendations. Got it? Not just recommendations, but this is a knowledge that you've got to share. And everybody, their research papers will be published on my Gifted Gabber website as well. It's still uploading it. Okay, so next up we have Shraddha. Shraddha is a writing senior from Medina, Ohio. Her topic is a deep dive into marine-derived drugs. Okay, so go ahead. Her passion is marine biology, as you can see. So go ahead, Shraddha.
Okay, I'll start screen sharing now.
Yeah, please go ahead.
Hey guys, my name is Shraddha Mandapudi. I am from Medina, Ohio. I'm a 12th grader at Highland High School. And what my research topic was a deep dive into marine-derived drugs, because it is a very new field of science that hasn't been researched until very recently. So for my introduction, I just had basically, what had started my interest for this specific topic was that marine-derived drugs have so much to offer, because the ocean's ecosystems cover 75% of the Earth, and only 5% of the oceans are currently charted and explored. So just like terrestrial-based derived drugs have so much to offer for cancer patients and for people who have other kinds of diseases, I figured that marine-derived drugs have just as much to offer. The question that I went in to my research was, are marine-derived drugs worth the time and money spent? And as you can see, once we go into it, the answer is very obviously yes. Some objectives that I had, some stuff that I actually wanted to find out, was that I wanted to identify drugs that use marine characteristics, and I wanted to examine these effects and uses of the drugs. So I compared and contrasted them to other drugs known to cure the same type of disease, or known to be used for the same type of problem. And I also wanted to determine the future uses of pursuing this marine pharmacology. For the literary review, I actually used this research paper in my research. I referenced it. It's Exploring the Ocean for New Drug Developments, Marine Pharmacology by Harshad Malv. And basically, again, it just goes into a more detailed explanation of other marine-derived drugs, and it includes a lot of classifications for marine pharmacology. It talks about a bunch of pipelines, and like classifications can really be talked about. Like marine pharmacology classifications are really just the basis or source of the candidate drug, either genetically engineered marine organisms manufactured of pharmaceutical or nutraceutical of marine origins, or chemical produced or found in marine organisms shown to have a wide variety of applications in pharmaceuticals. He also goes very deeply into pipelines for preclinical trial drugs. So I found this really interesting, and this had a lot to do with my current research. The methodology, because I was already researching like already discovered drugs or drugs that are being researched currently, and the only resource I had was the internet, I couldn't really do a lot. All I could do was look at the research papers and compare and contrast different drugs that are used for the same thing compared to marine based drugs. And really the only problem is, because marine pharmacology is such a new field, not many drugs are approved by the FDA, but the drugs that are approved by the FDA and are being used in hospitals, such as cytarabine, trabectedin, and ziconotide, all of which I have included in my research, have been extremely useful and powerful in fighting the disease. Cytarabine and trabectedin are both the most powerful drugs that are used in hospitals today to fight specific kinds of cancer. Cytarabine is used to fight off lymphocytic leukemia, and trabectedin is very useful for fighting off soft tissue sarcoma and relapsed cases of platinum-sensitive ovarian cancer. There are also other drugs that aren't approved by the FDA but are still very effective, stuff like painkillers. Ziconotide is a synthetic molecule that comes from the venom of a marine snail, and that is a very effective painkiller. However, it has not been approved by the FDA yet. So these are just the results that I had found. I had found that out of the 35 known drugs that are in preclinical trials or approved right now, only 10 of the drugs were approved by the FDA, which just amounts to a 29%, but the others range from painkillers to vitamin supplements, so they're still very useful. Just as a conclusion, again, due to the nature of my research question, the most I was able to do was go off from the internet and learn what I could from there. However, even with my limitations, I found that any drug that has been marine derived has been very useful in fighting off cancer specifically, or it's a very useful vitamin supplement, or it could be used as a painkiller. So even though that marine pharmacology is very new science, it is worth all of the time and money that has been spent on it, and that should be spent on it in the future. Thank you.
Very good, very good. We learned so much. Beautiful presentation. Guys, just a request that keep your presentation shorter. We have a lot of presenters. Got it? Next is Sai Bachcha. He's an 11th grader from North Carolina. His topic is the effectiveness of COVID-19 vaccine based on individual health and race. Okay, so Sai Bachcha, go ahead.
So I'm Sai Bachcha. I go to Marvin Ridge High School, and I'm an 11th grader. And I'm looking at the effectiveness of COVID-19 vaccines based on individual health and race. So basically, the main topic I'm looking at is the importance of a vaccine on an individual based on their health. So one of the main research questions I was looking at was which vaccine was the most effective and which one was the least, to make sure each individual is healthy when taking or proceeding to use the vaccine. So this could be a defining factor if an individual wants to take the vaccine in the near future. And some objectives I was looking at each clinical test trial to make sure that there was no fluctuation or correct values while taking the vaccine. So based on my literature review, so there has been a lot of research done on this topic, and the FDA has approved each vaccine and looked at it carefully. They've done multiple clinical trials to make sure that each vaccine is healthy and able to be used by individuals. And they have tables to showcase each race, age, and various other topics to make sure that each individual is correct. So basically, my results were pretty self-explanatory, as the Pfizer vaccine was the most suitable for human usage as it had the highest efficacy rating at 95%, while Johnson & Johnson had an efficacy rating of 66% and created a lot of tension between people as it caused various blood clots. And the most significant finding I found was the immense difference between Johnson & Johnson and Pfizer, as there should not be that big of a difference between vaccines. And in conclusion, the results go as followed: Pfizer, Moderna, and then Johnson & Johnson. And the contribution I made goes way beyond just breaking down the vaccine. I've also helped individuals understand which vaccine is best for their usage based on underlying health conditions and various other topics. So some of the limitations I had with the study included not being able to control who's tested in the clinical trials, and I couldn't control the outcomes or style of the procedure and how it was done. And basically, this is the effectiveness of COVID-19 vaccines based on individual health and race.
Okay, excellent, Sai. Next up, we have Anushka Jain. She's a 10th grader. Her topic is the role of emotion and social norms in human-robot interactions. Anushka, go ahead.
My topic was the role of emotions and social normatives in human-robot interactions. The introduction. This topic was AI and psychology related. AI and automations have been painted in negative light in the media far before AI technologies even started to exist. 43% of American consumers believe that if AI were to happen, it would result in more harm than good. It is important to highlight the lack of truth behind these presumptions and discuss the role of emotions and social norms affecting human-human interactions and human automation interactions. In order to conclude the role of emotions and social norms in human-robot interactions, we must first discover patterns of behavior within human-human interactions. After considering the nature of said interactions, we shall then apply such skills to human-robot interactions in order to create a sense of human trust and engagement with automations. Throughout this research presentation, we shall explore the role of emotions and social normatives in human-robot interactions. So here is my literature review in relation to the research. Past research and assumptions. Many research groups have been working on social and emotional cues implementation in automations, and many behaviors that further human trust in robots have hitherto been implemented. Past research done on human-robot interaction discloses that people address robots in the manner that the robot is addressing them. Therefore, the emotional functionality of the robot will impact the receiving interaction displayed by the human. Furthermore, if robots were to display the same basic behaviors as humans, a deeper level of human trust would be initiated. Different behaviors gathered from the human reading of such behaviors would make the robot seem unpredictable and frighteningly strange. We need to study the effect of emotions on human-robot interactions further. One might believe that increasing the robot's positivity when interacting with humans would increase its likability level. While increasing the amount of positive emotions robots show through emotional cues may increase the human's attentiveness towards the robot, it is important to study the effect of emotions as a whole on human-robot interaction. The methodology and results includes findings. Analyzation. The presence of emotions and social normatives is vital for technology to be deemed trustworthy to the user. Investigating the effect of emotions and social norms in human-human interaction, the effect of trust in human-human interaction, and the effect of trust in human automation interaction, we were then able to further explore the application of emotions and social norms in automations. Concluding from research, there is strong need for emotions and social normatives in automations in order to increase user trust levels and consequently user engagement and interest. Exploring the possible applications, in the future we may apply non-verbal cues, such as mimicry of human facial expressions and pupil enlargement, and verbal cues. If implemented, perhaps we may increase the worldwide trust level in automations, as well as improve consumer experiences. The conclusion, a post-deep dive into the topic. So restating our topic, the topic was the role of emotions and social normatives in human-robot interactions. The presence of emotions and social normatives is vital for technology to be deemed trustworthy to the user. I have contributed to this research on the Uncanny Valley, which, as can be seen in the figure, human likeness of a subject follows a dip based on familiarity. If we implemented such social and emotional normatives, we could increase user trust levels in automations and improve consumer experiences, which is our overall goal. Thanks for listening to my presentation.
Beautiful, guys. I just wanted to take a moment to thank Dr. Raj, who, he's like the architect for our program. He really encouraged children to dig deeper research. He gave them an opportunity to research about worm vaccine and everything. So he's not here. I just wanted to take a moment to thank him. Okay, so next up we have Kushal Bantu. He is he's a senior in O.J. Robbins High School. His topic is the mRNA COVID vaccine and its stability on the genetic level. Go ahead, Kushal.
Can you see my screen?
Yes, please.
Okay. All right. So like Coach Joe said, my name is Kushal Bantu. I'm a 12th grader at O.J. Robbins High School in Pennsylvania. And my topic is mRNA COVID vaccine and its genetic stability. So my topic of research involves the elements and processes of the mRNA COVID-19 vaccine. Some research questions I set out to answer was, what is the mRNA vaccine? How does it function? And does it actually affect my body's natural systems? And so the objectives of my study is to learn more about the vaccine's processes and inform others to eliminate confusion and fear. People still doubt the effectiveness and safety of the vaccine once it enters the body. So my study investigates the direct impact of the COVID-19 mRNA vaccine. So some research that I included in my paper involves the ingredients of the mRNA vaccine. I wanted to understand exactly what in the mRNA vaccine was so useful and so effective when people get vaccinated. Also, I included protein translation and transcription. These two phases are processes that take place in our cells to help create proteins. And the first phase is transcription. The second phase is translation. Transcription takes place in the nucleus of our bodies where DNA is located. And translation takes place in our cells, but outside the nucleus. So it does not interact with the DNA. And the reason it does not affect, the vaccine does not affect our DNA is because the vaccine itself has a component, the mRNA, which is actually the code that is created from transcription. So it does not actually interact with the transcriptional phase. It just goes straight to the translational phase. And so some theories that people have come up with are that the vaccine causes code variants, such as a Delta variant. This is actually false because what happens is the virus develops resistance as people are getting vaccinated, which causes the variants. And also, people think that the vaccine is reason for many fatalities, that because of side effects and such. Side effects can show that people are actually getting vaccinated because the negative effects in our body show that we're actually getting used to the vaccine. And so more research and understanding the virus is still required to better eliminate it because, the vaccine is still very influential today and it still has an impact on our society. And so what I have done is I have studied the methods and procedures scientists took to come up with the vaccine and the vaccine cellular functions in the body. And what I've learned is that there are 4 key ingredients in the vaccine: the mRNA, lipids, sugars, and salts. The sugars and salts serve as stabilizers in our body. Our bodies are always changing temperatures, changing environments. These sugars and salts are there to protect the mRNA so that the mRNA does not break down. There are also lipids in our cells or the vaccine that help allow the mRNA to enter the cells without breaking down. And the mRNA is the code, the active ingredient in the vaccine. So the vaccine enters the cell through endocytosis and uses mRNA and uses the mRNA to create these spike proteins. What these spike proteins do is they help produce these immune responses from our immune system. Our immune systems recognize these spike proteins as external substances, and they immediately start to create these antibodies to counter the spike proteins. And the antibodies are then remembered by memory cells, which will then be produced anytime anyone is ever exposed to the virus. So there's an immediate response if anyone is ever exposed by the virus. And so my most significant finding is that the vaccine creates this immune response, which produces the antibodies. And it does that without changing the DNA or genes in our cells, which I said before is because it does not include the transcriptional phase, it includes the translational phase. And so in conclusion, my main topic in question again, what is the mRNA COVID-19 vaccine and how does it impact the body? My purpose or contribution is to learn and teach other people about the vaccine and its properties to eliminate uneasiness and fear when getting vaccinated. Also, I intend to influence others to get vaccinated to eliminate the virus's effects. I know everyone wants to return to their normal lives, so obviously everyone wants to get vaccinated. And one limitation is that there's still a limited amount of access to information about the virus and the vaccine itself. However, my research still enforces the importance of the vaccine and its impact on the body. Thank you.
Very nice, very nice. Next up, we have Atish Vats. His topic, he's a 10th grader in Garnet Valley. His topic is how lead affects the brain. Go ahead.
Okay, so as Coach Joe said, my name is Atish Vats. I go to Garnet Valley High School in Glen Mills, Pennsylvania. And my topic is how does lead affect the brain? So the topic I am studying is lead and how it affects our bodies, the brain in particular. The research questions I set out to answer was how lead enters the body, how it moves through the body, and how it affects the brain. I was attracted to this topic because my brother has lead poisoning, and I want to learn more about the topic so I can help my brother. So about lead, there are several articles and research papers that have been written on how lead affects. Here's some examples I came across in the course of my research. However, my research is not perfect and there are some errors. One area of weaknesses in my research is that there is so much about the brain that we still don't know, which needs more research and studies to help understand how lead affects those parts of the brain. So when I analyzed the data, I found that lead enters the body through inhalation and ingestion as showed in figure 1, or in other words, you can eat lead or breathe in lead. Like, for example, little children like to chew things, and some of those things might contain lead. And also, you can breathe in lead. For example, when you walk by a construction site, you can breathe in lead dust. Lead moves through the body via the bloodstream, and it affects the brain by mimicking the role of calcium in the body. So the most significant finding in my data that lead can affect the prefrontal cortex, which is responsible for attention, complex decision-making, regulating social behavior, as shown in figure 2, which is MRI scans of the brain, and it can narrow the blood vessels, which forces the heart to work harder and can potentially expose it to a heart attack. So the question posed in my research paper was how does lead enter the body, how it moves through the body, and how it affects the brain. My findings were again that lead enters the body through inhalation and ingestion, moves through the body via the bloodstream, and affects the brain by mimicking calcium. Hopefully, my research has shown the vast dangers of lead and how the body can help parents prevent lead from getting into their children's bodies, and hopefully help educate people to the dangers of lead. Okay, I'm done.
Beautiful, Atish. Very, very nice. A very nice job in educating everybody about lead and your research. As you can see here, these kids spent most of their summer doing research. So they are going to answer the question in their college admissions as to what they were doing in summer. And research is what they were doing. They really worked hard, dug deeper, and being in this group is not necessarily easy. So they did a fantastic job. Next up, we have Ritisha Suresh. She is a rising senior in Seminole County, Florida. Her topic is peptide immunotherapy for the treatment of food allergies. Go ahead, Ritisha.
So hi, everyone. Like Coach just said, I am Ritisha, and I am in 12th grade, and I go to Seminole High School in Orlando, Florida. So my research question was, to what extent can peptide immunotherapy be considered the leading novel curative treatment for food allergy? not sure how many of you know, but food allergy significantly impacts around 18% of the world population, posing lifelong health risks that are often untreatable. Currently, the only treatment for it is to avoid allergens and to always carry around an epinephrine injector. It does not solve the problem of an adverse immune response, which should be the goal at this point. Peptide immunotherapy is one of the novel curative treatments that have arisen. It aims to induce desensitization or tolerance, or lack of an immune response when an allergen is present, making it an extremely important part of medical research. It also lowers the risk of severe side effects typical in food allergy immunotherapy, making it novel. My research aimed to evaluate the efficacy of peptide immunotherapy in comparison to other novel treatments and also treatments that existed given current research. So current food allergy immunotherapies that do exist right now are oral immunotherapy, epicutaneous, and sublingual immunotherapy. These three have been researched and have been proved efficacious, but they do have their drawbacks, such as a high risk of IgE-mediated responses, which is just a subset of an immune response that you can get from food allergy. The cost, there is a peanut oral immunotherapy called Palforzia, and that costs around $4,200 per patient, which is extremely expensive. And it only induces desensitization, not tolerance. There's a difference. Desensitization is just making sure you don't react to the food allergen, while tolerance is making sure that the allergen can actually be reintroduced into the diet. It is something that you can tolerate and eat. Novel food allergy immunotherapies that are currently in research and testing are peptide immunotherapy, adjuvant immunotherapy, and DNA vaccines. Since I focused on peptide immunotherapy, some of the drawbacks of that would be that they have to be done on a case-by-case basis due to the hyperspecific allergens identifying T cells. There's needle aversion because peptide immunotherapy is a vaccine that you have to administer every so often, and it might not be applicable with for some with pre-existing conditions. The methodology that I used for this was just that I agglomerated research to determine the efficacy of PIT. It was then compared to existing food allergy treatments along with 2 other novel treatments that I discussed previously. In terms of efficacy, it has been tested largely on mice and rat models for common allergens such as egg, peanut, and cow's milk so far, and most of these results were statistically significant and provided support for human clinical trials. For example, peptide immunotherapy was actually found to induce tolerance in mice tested with peanut allergens. However, PIT's efficacy in comparison to the efficacy of DNA vaccines and adjuvant IT is unclear, as comparative studies have not yet been conducted in this realm of research. So after research, I've concluded that PIT's current practical usage as the novel curative treatment for food allergy is not definitive, but it does have a myriad of research, theory, and preliminary results that postulate large-scale future use. And my contribution to this is the fact that it evaluates the use of PIT in food allergy treatment and provides accurate information that highlights its advantages and potential in the medical field. Thank you.
Bravo. I hope you continue this research. Both my sons had food allergies and I used to carry EpiPen everywhere. I've administered it twice as well. Okay, so next up we have Sanjana. She's a rising senior from Rhode Island. Her topic is very interesting. It is, I hope I don't butcher it, is aducanumab, the miracle drug. She's going to talk about it. So I'm sorry I butchered the thing. Yeah, go ahead, Sanjana.
Can you all see it?
Yes, go to the first page and then start presenting. Yeah.
Hello, everyone. My name is Sanjana Anantola, and I'm from Rhode Island. And my research topic is aducanumab, which is a treatment for Alzheimer's. One of my interests is neuroscience, and when reading, I stumbled upon this article discussing aducanumab, and after reading, there were so many questions that I had. And so for this research paper, I felt that I could fuel my passion for Alzheimer's disease by learning more about aducanumab. Here I've listed my topic, some questions I had when reading, and my objective, which was to understand more about this drug and learn about the progression of Alzheimer's disease. Moving on to the literature review. Let's start off with Alzheimer's. This was a common topic that came up, well, obviously because we're treating Alzheimer's disease. So Alzheimer's disease is a neurodegenerative disorder where there is a dominant negative genetic mutation in a patient's 21st chromosome. As you can see in the picture, the brain decides to shrink and the ventricles enlargen. And this is very harmful, which means because the neurons are dying, and that means cognitive functions aren't as well or good as they were before. And down here are some scary statistics, as well as some symptoms that are common in Alzheimer patients. Another thing that came up was amyloid and beta and tau protein, and Alzheimer's disease is caused by the clumping of the excess of tau and amyloid beta protein. And these clumps create plaques within and outside the neuron, limiting the cell's ability to properly function. And here we can see several pictures of how it happens and the dangers of why this clumping is really harmful to the brain. Before aducanumab, many of the other drugs were considered to cure or to treat Alzheimer's, and these were called chlorine esterase inhibitors. These inhibitors would focus on promoting neurotransmitters instead of dealing with the plaques. However, immunotherapeutics use antibodies to alert the immune system of possible threats. Here we can see a picture of how immunotherapeutics are used in cancer. And so a tumor cell is there, and then that drug produces antibodies, and memory T cells they see the antibodies and they are able to go and attack the tumor cell. Keeping immunotherapeutic procedures in mind, we can understand how aducanumab works. Monoclonal antibodies are attracted to peptides in the amyloid beta, and once they attract, the antibodies mark the plaques. Then the B lymphocytes produce T cells to help destroy the clumps. Moving on to the clinical trials, aducanumab has been tested on 1,105 patients, while the rest were given a placebo. Of these 1,105 patients, reports say that approximately 52% were female, 76% were white, 10% were Asian, and 3% were Hispanic or Latino. So there's a large diversity and different race, so aducanumab was tested for all people. So what are the results? From these graphs, we can observe that it's clear to see the decrease in amyloid compared to the values of the placebo. 3 studies were done and all gave out similar results. In conclusion, aducanumab seems to be successful. Here are some specific examples of what we noticed in the graph, such as the change in week 26. And we can see that the solid line was the placebo test. It always stayed constant, and sometimes it increased, but all the different dosages with aducanumab made it decrease, which seems to be successful. And through this research paper, I have given more people information about this new drug, and such as how it works and its success rate. Recently, a new controversy has come up with Alzheimer's disease, and it questions the functionality of this drug. Either way, I still believe that aducanumab is a miracle drug because of the change in the progression it has created, since it's the only drug that has addressed the plaques. Thank you.
Very, very nice. As you can see, how exciting my job is. I mean, day in, day out, I come here and I learn new stuff. That's how wonderful my summer has been with your kids. Okay, next up, we have Isha Lingala. She's a 9th grader from Nashua, New Hampshire. Her topic is analyzing toxicology reports of the COVID-19 vaccine. Go ahead, Isha.
Hi.
Yes.
Can you see my screen?
Yes.
Okay, so hi, I'm Isha Lingala. I'm a rising sophomore at Nashua High School South. And my thesis is on analyzing toxicology reports of COVID-19 vaccines. Okay. So the main points that I make in my article are the following. The S protein is crucial for the infection of the virus, and without it, viruses would not be capable of reacting to potential host cells. As such, it serves as an ideal target for treatment. Recombinant vaccines function by instructing host cells to assemble the protein of the S antigen distinctive to SARS-CoV-2. By creating the protein, memory immune cells retain the necessary information and create an immune response to the virus. DNA and RNA-based vaccines fall under the nucleic acid-based coronavirus coronavirus vaccines subcategory, which belong to recombinant vaccines. The animal models have all shown incredibly successful results in the clinical trials. Live attenuated vaccines and inactivated vaccines take a different approach than recombinant vaccines. Live attenuated vaccines use weakened live coronavirus, and inactivated vaccines use the inactivated virus, which means it still contains the target virus, but it no longer has its nucleic acids and its antigens are intact. The results yielded are the following. The efficacy and development of the vast subcategories of recombinant vaccines, especially mRNA coronavirus vaccines, is superior to that of live attenuated and inactivated vaccines. All vaccines deal with opportunities and obstacles, yet recombinant vaccines utilize an appropriate target, the S protein, and provide the most effective treatment. mRNA vaccines do not necessitate access into the nucleus of the host cell in order to be transcribed. mRNA vaccines, like most recombinant vaccines, instruct cells to make a protein which then goes on to trigger an immune response. They also have the benefit because a lower dose can be used without the need for any special delivery mechanisms. Moreover, mRNA-based vaccines avoid the risk of integration with the host cell genome and are able to produce pure viral protein. I conclude my question of, is one type of vaccine superior to the others? With a concrete answer. Recombinant vaccines, especially mRNA coronavirus vaccines, are superior to live attenuated and inactivated vaccines. Further research into many safety concerns and as well as further clinical trials on humans must be conducted, as most clinical trials were done on animal models. I realize I think I skipped my introduction, but yeah. Thank you.
Very good. Next up, we have Samanyu. He's 11th grader from Coles Neck, New Jersey. His topic is overview of ADHD and what genes affect it. Samanyu, go ahead. Yes.
All right. So, hi, my name is Samanyu Kashyap, and I'm an junior in Coles Neck High School. And today I'm going to be talking about a comparative study of ADHD and how ADHD medicine affects your brain, as well as some of the genes that affect ADHD. Now, so what is the actual topic area that I'm researching? So the topic area that I'm researching is the overview of ADHD, as I've said, but more importantly, it's some of the least talked about aspects of ADHD, such as the genetics or the medicinal aspects. Now, what are some of the specific questions that will be answered? And some specific questions will be like how it actually affects your brain, like physically, like what happens, like what causes you to be hyperactive, what causes you to lose focus, as well as what genes cause ADHD, like what is the underlying cause of the hyperactiveness, of the loss of focus. And what was the objective of my research? And the main objective of my research was to kind of explore the least talked about aspects of ADHD and kind of bring more attention to them, because I personally feel like these aspects of ADHD aren't talked about enough. And kind of a secondary objective of my research was I also suffer with ADHD, and I kind of wanted to kind of research my own kind of, I guess you could call it a problem, my own kind of thing, and kind of understand myself a lot more better. And I also take medicine for ADHD, so kind of understanding what I put into my body every day, it's kind of what drove me to research this in the first place. So basically, like some of the relevant research that is close to my current topic was regarding the genes for ADHD. So basically that it talked about how the genes for ADHD, it runs in twins a lot more. So there's the D5 receptor gene and the D4 receptor gene. And these are the two most possible candidates for the candidates that cause ADHD. Now, both of them have lots of evidence for and against those genes. So it's not really kind of like clarified which gene is the actual true gene that causes ADHD. It's not even clarified if it's one specific gene that causes ADHD. It could be a combination of both of them, but those are the two kind of main genes that are supposedly causes ADHD. Now let's go over some of my methodology and results. So some of the most significant findings were that ADHD medicine affects your brain but increases the amount of dopamine, helping concentration and reducing hyperactivity. So basically what happens is in ADHD, people who have ADHD, they have an increased amount of what's called dopamine receptors. Now these limit the amount of dopamine that gets into your brain, which increases the hyperactivity levels. Now what ADHD medicine does is it decreases the amount of dopamine receptors, allowing more dopamine to get into your system, which therefore reduces the hyperactivity and increases the focus. Now, ADHD medicine also increases your level of norepinephrine, which is a chemical that is associated with increasing your heart rate, and it basically makes you feel excited or like anxious. Which is like, as you said, like increasing your heart rate. So that also ties into ADHD, like the hyperactivity and the lack of focus. Now, regarding the genetics of ADHD, as I said before, there are two main causes or two main candidates, which is the D4 and D5 receptor gene, and they're both as equally to be the cause for ADHD. And as for how I actually did my research, most of my research I found from PubMed or like actual research papers. And I also talked to my neurologist about ADHD and some of the symptoms and the medicine that she's been giving me and all that kind of stuff. So I talked to her like more in depth because there's some information that you can't really just find online. You really have to talk to an actual person. And that really helped me with finding some of the information in my paper. So now let's get to the conclusions. So I really feel like some of the contributions that I've made to this kind of field is I really feel like I've accomplished my goal of bringing attention to the least talked about aspects of ADHD, but at the same time, extremely important. Like not everyone takes medicine for ADHD. You only take it if you really, really have to. And the dosage you take is really important. Like in some cases, if you take too high of a dose, people are reported to have almost zombie-like rabies symptoms. So I really feel like these are the most important aspects of ADHD, but people really don't talk about them enough. So by doing this research paper, I really do think that I've accomplished my goal, which I set out in the first place, which is to kind of bring more light, bring more attention to these aspects so more people can talk about them and more people can get answers to like what is causing this and what is like the underlying cause of this. Like ADHD is not like a rare illness. Like 5 to 9% of Americans suffer from ADHD. Like it is not a rare illness in any way. So I really feel like bringing more light to this subject is extremely important. So thank you for listening to my speech.
Very nice. Very nice. You guys see the passion in these people. They're either trying to solve the problem that they are encountering or their siblings or around them. Gen Z gets a lot of flack for not being serious or being addicted to video games or not having that kind of attention span. But in my opinion, the Gen Z generation is far more intelligent than any others, and they consume information in bite size and they're super intelligent. So great job, Samanyu. Next up, we have Aditya Mahajan. He's a rising senior from Dublin, California. His topic is developing a deep learning approach for skin cancer multi-classification using the HAM 10,000 data set. Go ahead, Aditya.
Hello, can you hear me and see my screen?
Yeah.
My topic is using deep learning to classify skin cancer. So the topic here is, can we use a deep learning approach to classify various kinds of skin cancer? And can we write an algorithm to do this for us? The objective for us is to write an accurate algorithm to classify specific types of skin cancer. So first, I had to read some other papers that did similar things. So research has already been done with published models that get close to a 90% accuracy on the HAM 10,000 data set, which is the data set that I will be using for my own model. The HAM 10,000 data set is really useful because it has 10,000 well-took dermatoscopic images that are all labeled with the type of cancer. And these other models usually use ResNet. ResNet are a kind of model that I would have used, but I couldn't use because I didn't have a device with the proper CPU. So instead, I had to use a traditional CNN. And so with the ABCDE signs of skin cancer, which is over here, asymmetry, diameter, colors, exposure, and evolution, I was able to write a program that looked for these specific things and how they change between the different kinds of skin cancer. With this, I was able to make a deep learning model using Keras, a CNN model using Keras with TensorFlow, which is a Python library. And I was able to attain 80.9% test accuracy and 0.339 low. And so here I have two graphs. This one with accuracy between the training and test set, and this one with model loss between the training and test set by epoch. And an epoch is just one running of the model. So each time the model is run, some parameters are tweaked, and then the model improves. And here is a flowchart that describes the model's general method of classifying a specific skin cancer. So we were able to meet our objective in creating an algorithm to classify skin cancer, since we were able to get an 80.9% test data accuracy and 0.339 model loss. Now, some benefits to the research community from this project is that this model will be available on GitHub as an open source project. So anyone can see how we made our model, avoid any of the mistakes that I made, and use my model to make their own better, or they can even add on to my model. Now, some limitations with using deep learning to classify skin cancer is that not all models work with all data sets. I was lucky because the ham 10,000 data set, was super clean and it was ready to be used. Normally, cleaning out the data set is a process that takes hours. And not every clinic has the access to the right technology to be able to do this. And thank you. That's it for my presentation.
Nice. Like deep learning and AI is something that is getting more and more popular in the COVID-19 industry. And
so my name's Trishon Swapnanandan, and I'm a rising sophomore, and I did my research on the use of artificial intelligence, deep learning, and machine learning in fighting COVID-19 and vaccine development. So the research question I answered was the effect artificial intelligence, deep learning, and machine learning had in fighting COVID-19 and vaccine development. Some of the questions I answered were the correlation of AI and big data, whether the effect of AI was positive or negative, and to analyze the different aspects such as deep learning, machine learning, as had a direct correlation with AI. As the COVID-19 infection spread to 212 countries, researchers were struggling to handle the ever-growing big data. AI is one such technique that deals with big data using deep learning and machine learning, and this bridges the gap between science and technology. So using machine learning and deep learning technology, we understood the pattern of viral spread and quickly analyzed the big data, and this helped understand the characteristics of COVID-19 and develop drugs, and which eventually led to vaccine development. So these are two important images that I wanted to explain. So in this first image here, when the pandemic started, epidemiological data, clinical data, and genetic data were managed and processed using AI, machine learning, and deep learning, which helped better prevention, better hospital operation, helped with diagnosis, and got more therapeutics. In the second image here, this gives a brief explanation of artificial intelligence, machine learning, and deep learning. So artificial intelligence is a technique which enables machines to mimic human behavior. Machine learning is a subset of AI technique which uses statistical methods to enable machines to improve with experience. And so deep learning is a subset of machine learning which makes the computation of multi-layer neural network feasible. So these are my methodology and results. So AI systems can learn more from successive cases and be exposed to multiple issues within minutes, which far outnumbers the cases a clinician could evaluate in one mortal lifetime. Some of my significant findings are AI usage in medicine not only reduces manual labor and frees up the physician's time, but also boosts productivity, precision, and efficiency. My conclusion is that AI is the intelligence demonstrated by machines that show cognitive behavior similar to humans. My analysis shows artificial intelligence has a positive impact on COVID-19 in vaccine development, helping contain and understand big data. My contribution is to bring some insight and knowledge to everyone that reads my research paper and help them understand the impact of AI on the COVID-19 pandemic. Thank you, and here are some of the links I used.
Yeah, very nice, Roshan. Very interesting topic. AI and machine learning was almost helped a lot with the vaccine research and everything. Very good. Excellent.
Thank you.
Next up, we have Ronit Bokison. He's from Trinity Preparatory School in Florida. His topic is effect of COVID vaccine on the development of tachycardia. So you're going to learn about that topic, tachycardia. Go ahead, Ronit.
Okay, so my topic is the effect of COVID-19 vaccine on the development of tachycardia. So this topic has already it's newly introduced, and it's regarding cardiovascular effects on the COVID vaccine. And I wanted to find out why the heartbeat had a major problem the heartbeat was affected majorly after a vaccine injection. So that's one of the main reasons I chose this topic. So some current research done on different underlying heart conditions from the COVID vaccine has been conducted by the CDC. And this is very, very rare, only 1,000 cases, which is like 0.00059% of all vaccinated people in the US. And some theories were from a doctor from Malaysia, and he stated that tachycardia increase is subject to the population of older Americans. So what this researcher's theory is basically, since older people have more underlying heart conditions, that can lead to excess tachycardia after the injection. So my methodology and results. So here is a figure of the heart variation after doses of COVID vaccine. So basically the vaccine contains an mRNA strand, and this mRNA strands fight against the SARS-CoV-2 spike protein. So basically my theory is that that COVID vaccine is produced from the SARS-CoV-2 because immunological responses happen when the mRNA meets with the SARS-CoV-2. So basically the immunological responses create an increased heart rate as proven by many doctors. Even when taking shots such as the flu, when fighting immunity, you will feel sick and an elevated heartbeat. So some of my conclusions. So as one of the vaccines is the Pfizer vaccine, and this has a nucleoside modified mRNA from the virus's genetic coding, and this virus from the SARS virus fights against the nucleoside modified mRNA, which changes and alters the protein in cells, and this allows the body to have more immunological responses and resulting in POTS illness. And some of the limitations on this study were because this is a rarely studied vaccine, and this is only 1 or 2 cases ever were produced on this. So more research needs to be done regarding tachycardia's effect on the vaccine. And some contributions I made was I created some graphs regarding the mineral and vitamin levels of people and patients with tachycardia compared to an average male. And I helped insight towards the impact of the COVID vaccine regarding underlying heart problems. And that's basically it.
Very good, Ronit. Okay, next up we have Anish. He's a 10th grader from Michigan. His topic is current statistics, prevention, and pathology of COVID-19. Okay.
Hello, everyone. My name is Anish Komati Reddy. My topic is the current statistics, prevention, and pathology of COVID-19. I am a rising sophomore, and I'm in Michigan attending Novi High School. So my introduction. My main topic area is the pathology of COVID-19. I have a couple of questions that I'm going to answer now and later in my presentation. So my first question is, what is the pathology of the virus once it's inside the human body? The second is how can we educate people on why they need the vaccination? So the first question I'd like to answer is we would help the world better prepare for upcoming viruses and diseases. The second is to mitigate the effects of COVID-19 and prevent possible virus outbreaks. My main objective is to understand the disease's pathology and why people need to be vaccinated and maintain precaution. And so this is my literature review. And since of March of 2020, COVID-19 has had a 76% growth rate. This chart displays the growth of COVID-19 within each country and how it has spread and immaculately grown throughout the months. So this was a chart that was created by me using a tool online. I've gathered all my resources from the FDA, PubMed, Google Scholar, and other sources I found online. And I've made a chart varied from each month on how COVID has been growing and increasing in numbers. So if you guys were to look at 3/16/20 on the United States, it would start at about 4.6K of infection cases. But once we move on to 3/19 of 20, then it would be to 13.6% of people, which was a 76% growth rate, which was extremely bad considering how little protection we have done over the past few months. So this is my methodology and results. And so below, I've made a chart using different sources that represented the growth of COVID-19 in different countries and the size of its growth by each month/year. I then turned all my data using the sources that I've collected as shown in the previous slide, and to show the different levels of peak infection rates for various countries. So if you guys were to look in the graph, you could see that the USA has a very sharp increase in death/infected rate, and with mainland China having like a plateau effect, which is kind of like curving back and going on to like a slope or steady value. And so the results that show here will continue in the numbers of COVID cases, but unless there's a secondhand disease or another variant that is affecting COVID-19 will still continue and rise in cases. And so one note is that countries also need to be aware that even with the provided resources, they can still be affected by the waves of COVID-19. And so what I mean by this is that recently some news has come up that there is a COVID Delta variant. And researchers and scientists have been saying that you would need a booster vaccine for the next 6 months. And this kind of proves my point in saying that even if we were to take 2 of the vaccines that helped us for the first year or so, we would still have to take a couple more to wear out the effects of boosters or Delta variants in the COVID-19 infection. And so my conclusion is that, just as a side note, information is changing day by day. So my prediction based on one day could change the next day and alter and succumb to different data. So what I mean by this is, for example, 1 week COVID might have a downside effect is that not many people are being infected as supposed to be, and the graph could sometimes go down even. But then the next month, it would have a sharp spike and people could be double or triple being infected by COVID. And so in conclusion, I believe that there are subtle but significant ways we can fight off COVID-19. As it is a high infection and transmutative virus, some of the methods include wearing a mask, maintaining at least 6 feet of social distance between one another, and learning the pathology of the virus, as this will decrease the severity of COVID-19. One benefit to knowing the virus's pathology is understanding what impact it has on the human body. COVID-19 has direct pathway to the vital organs, such as the lungs, liver, and all respiratory systems. In the end, it depends on what we can do as a whole community, how we combat the virus, how we can save lives, and how we can prevent future generations from succumbing to the likes of deadly viral infections. Thank you.
Very nice, Anish. Very good use of statistics and everything. So like students were asked to come up with their own statistical data in their research paper. That was their contribution. So great job there. Next up, we have Rishi Dev. He's a rising senior from Katy, Texas. His topic is the quantitative research employed by vaccine developers.
Hi everyone, as Coach Joe introduced me, my name is Rishi Dev. I go to Tompkins High School in Katy, Texas, and I'm a senior. And my topic is the quantitative research employed by COVID-19 vaccine developers and its approval by the FDA. So without further ado, let's get started. So the topic area that was addressed in my research was biotechnology, and to be more specific, it was driven on the research aspect of COVID-19 vaccines. And the big picture questions I set out to answer throughout my research process was: one, was the FDA too quick to approve of the COVID-19 vaccines' distribution? And secondly, did vaccine researchers accumulate the required data and research that proves the existence of a safe COVID-19 vaccine? And an objective that I contained within my research, aside from all the big picture questions I looked to answer, was to highlight the safety of these COVID vaccines. So literature review, there's a lot of research that ties in with the creation and distribution of COVID-19 vaccines. And for starters, is the theory that there's an inclusion of microchips in COVID-19 vaccines, which is basically a widespread phenomenon, especially here in the United States, that the government is looking to track, is insert microchips into the COVID-19 vaccines in order to possibly track the citizens of the country. But a lot of research, predominantly most of the research, has proved this to be false information and continues. But this debate continues to be a widespread phenomenon. Secondly, is warm vaccines, as I addressed in my research as well. Warm vaccines are vaccines that can be carried throughout any temperature. Especially can go up to 100 degrees Celsius, which makes it very effective for people to get vaccinated, especially on the go, because usually normal vaccines such as mRNA vaccines and Pfizer and Moderna, actually to be exact, they're required to be held at an exact temperature, so it's really hard for them to move the vaccines without limiting its effectiveness via the temperature. Third, it's can infants get vaccinated? So big picture is that since people only that are 12 and older can get vaccinated, especially with Pfizer and Moderna, researchers that I've been into maybe expanding that age limit to younger kids in order for them to get vaccinated as well, and thus boost up vaccination numbers in the United States. And that continues to be a widespread phenomenon. And lastly, this can be taken as both a tied-in research as well as a counter-argument, is the failure of the Johnson & Johnson vaccine, or at least the FDA didn't approve of the vaccine due to the side effects that it gives to many people, such as blood clots. And many people argue that this is also an attempt by the government to, potentially harm the citizens over giving them an effective vaccine. So methodology and researches. So what I've proved with my research is that both the Pfizer and Moderna vaccines have gone through rigorous clinical testings, and through these clinical testings, they have proved that not only are they safe and effective, but they can also be taken with other vaccines within a certain amount of weeks or days. And secondly, I also proved in my research that numerous mutated strands fall under the target reach of these vaccines. So not only do this original SARS-CoV-2 or Delta variant or MERS-CoV, all these variants can basically get tackled on by Pfizer and Moderna, which makes it really important for people to get vaccinated throughout, because it gives a challenge to the virus and its effectiveness. So to wrap things up, again, my research question was the quantitative research employed by COVID-19 developers and its approval by the FDA. And what I proved was the effectiveness of both Pfizer and Moderna in the United States. Like I said, through their clinical testing, through their approval to the FDA, it kind of sums up my big picture questions as to did the FDA rush a little too fast in order to approve these vaccines, which obviously ended up being no, due to the plethora of research that vaccine developers regarding COVID-19 accumulated. And to address some limitations of my research, it can include vaccines cannot be distributed to younger children, which, again, like I said before, there's more research being developed and gathered in order to tackle this issue. And lastly, research on vaccines was based only in the United States, so my research is only speculated and addressed in the United States. But with further research, many countries have also attempted to obtain Pfizer and Moderna in order to distribute it to their people in their countries. So thank you guys, and stay safe.
Thank you very much. Very good. Next up, we have Riya Ravindran. She is a rising junior in Princeton High School. Her topic is the effect of COVID-19 on patients with sickle cell disease. Go ahead. Yeah.
And my name is Riya Ravindran. I'm in 11th grade, and that is me.
Okay. So my main topic is how COVID-19 affects patients who already had sickle cell disease. And more specifically, I wanted to answer questions such as, does COVID-19 heighten certain symptoms of sickle cell disease, such as like body pains or certain like organ failures or etc., right? I also wanted to answer if certain symptoms of COVID would show up more severely or more often within these sickle cell disease patients than those patients without SCD. And you can see in my other slides, you can see the research really dives into this question, the second one the most. And I also wanted to go into whether COVID in sickle cell disease patients has a higher mortality rate or not. So the reason I chose this topic was because I think that, I don't know why, but for some reason, sickle cell disease has always stuck with me since I learned about it in like 8th grade. I thought about it, and I think before we started the research class, I learned like randomly that it is like way more present in Black Americans than in any other race. And then I also learned that COVID is the same way. So then I was like, let me just try to find a connection between these two. And then I researched about it, and there's a very much a connection. So the medical inequalities really just interested me. And this is a little genetic chart that shows how it's passed on. So if this parent has one sickle cell disease trait and one non-sickle cell disease trait, and the same with this parent, they're likely to have 1 in 4 out of their children who has the disease. And these two will carry it and they can pass it on to their children, but they won't exhibit signs of it. Okay. And this is just a picture explaining sickle cell disease. So pretty much like there's a blood cell that's a circle usually, but there's a rod that kind of forms inside the blood cell and causes it to fold. And so the main surface that holds oxygen on it to deliver it throughout your body and from the heart and back, right? From the lungs and back, sorry. It can no longer really hold it effectively, and it clumps up and causes major problems. So current research that is related to my topic, right now, sickle cell disease, they're finding a lot more treatments and drugs to help with the effects of it because of the body pains that are so severe. So hydroxyurea, I don't know how to pronounce that, is an example. And also patients with like very intense respiratory failure because of COVID-19 can also now use dexamethasone, which pretty much like slows down the breaking down of the lungs. In my project, I had a few areas of weakness because some of the studies just didn't have like a conclusive answer to some of the questions at the end of it. Like one of them, it did not really explain why the death rates of these patients was statistically at the same level of people who didn't have SCD. And they said that it would be because the people with SCD had intense hospital care, but we aren't really sure about that. So this is my first study, and pretty much overall, I just examined studies done on patients with SCD versus patients without SCD who contracted COVID, and then I made charts for them, and I examined the results, and then drew conclusions from them. So in the first study, it had 122 patients with SCD and then 172 with just a sickle cell trait, and then they compared it to about like 15,000 Black patients without any form of this. So they compared like a small number to the general public, but then they like used statistics to make it the same statistically. And so this is my chart for that. And it pretty much found that there were a lot more symptoms that were present in SCD patients as I expected. Hospitalization, body pain, pneumonia was much higher in the people with SCD than without it, as you can see in the percentages. So then in this study, this is a second study, they had 370 COVID patients who had SCD and found that they were 6.2 times more likely to die than the general population of Black patients who contracted COVID and did not have SCD. So this is the age ranges for that more specifically. Yeah. So less than 1% of them died, but then here it's way higher, especially in this age range. And they also found that young adults and children were more likely to pass away with COVID if they had sickle cell disease. So then in my third study, this was a registry. This one I found the most interesting because people would actually put in their own information into here. So of course this one, we can't really find it to be like fully, like reliable, but it is like, it's backed up by hospitals, but still there could be like mistakes in the entry information. So here it just talks about disease severity within this registry. And so you can see like it mainly lies between mild, moderate, and severe when people had COVID and sickle cell disease. So yeah, so my conclusion, once again, I just was asking how COVID affects people with sickle cell disease through the symptoms, the severity of the disease, and the death rates. So I think that my contribution was that I examined like multiple studies and then found conclusions from them that also connected to one another. And I think that it also is important to talk about these and to have a good like an overall conclusion with it, because it's important for people who are currently in the COVID situation who have sickle cell disease to know that they must be safe, especially since like they are at high risk. So yeah, and also there may be more of this research with the Delta variant coming up, and I also contributed with charts. So I can't control like the certain things I talked about before, like how the experiments were done and etc. But yeah, that is my project. Thank you.
Yeah, very good presentation. And the actually students were supposed to be only present, but then I mean, who cares about the time? They're so passionate. They're showing their work. So I'm sorry if it's over the time limit, but I'm just enjoying this. Okay, seeing them presenting all their work and their hard work. So next up we have Arushi Biswas. She's a rising junior in Westminster School District, and her topic is how neurons affect the well-being of humans. Go ahead.
Hello, my name is Arushi Biswas, and today I will be discussing how neurons affect the well-being of humans. I am a rising junior at West Windsor Plainsboro High School North in New Jersey. So my main topic that I will be discussing today is neurons. If you don't know what neurons are the fundamental units of the brain, and they are responsible for practically everything you do in your daily life. So the main questions I want to talk about are how can we enhance neuron function, and how do neurons function in a disorder called ASD or autism spectrum disorder. Now, the reason why I chose this topic is because of my brother. He has autism, and I was just interested in what makes autistic kids different from the norm and what we can do to help them. So the main limitations in my research were that there were not many studies on autism and different things that can help it, but I used whatever research I could find to draw my conclusions. So there were studies on something called essential oils, which have been shown to be greatly beneficial. And there were also studies on the effects of activities such as meditation and doing drugs. However, these activities, and some of them are not as beneficial, like drugs. They will cause a surge of dopamine or serotonin, but since these drugs are the ones that are triggering the release of dopamine and serotonin, the body's gonna constantly ask for more. So, as I said, my methodology for gathering results was after I analyzed a study that was done in Taiwan about aromatherapy and a study on jasmine oil. And this allowed me to hypothesize whether essential oils are beneficial for not only just everyone and the general public, but for kids that suffer from autism spectrum disorder. More specifically, kids with autism that tend to be more hyperactive and have a short attention span. And from my results, essential oils did show to have positive effects on these people's well-being, as they can relax and calm their mind, it can cool down their nerves, and it can improve their quality of sleep. And my most significant find was for ASD. One thing to note about autism is that not all kids with autism are hyperactive or have short attention spans. But for kids like my brother who does suffer from this, it can benefit them really greatly. So for my conclusion, what was my research question? How is well-being able to be improved for neurons to function properly? Well, there are many ways for better well-being, and I found research in essential oils, which can greatly help us. But as there was limited research in essential oils, I had to derive my own conclusions. And I think that there should be more research going into essential oils and how they can help us and disorders such as autism. And yeah, that's it.
Bravo, Anushka, research. And as you can see, some of the research topics are deeply personal to these students. Either they struggle with it or they have siblings who struggle with it. So it has been a wonderful journey working with all these kids. Okay, so next up we have Shrikar. He's a rising senior in Hillsborough School District in New Jersey. His topic is comparative study on the pharmacology and toxicology of 4 COVID vaccine. Go ahead, Shrikar. And guys, please answer the question in the chat and please ask questions.
Okay, so my name is Shrikar Srinivasan. I'm from Hillsborough High School in New Jersey, and I conducted a comparative study on the toxicology and pharmacology of 4 different COVID-19 vaccines. So this is my introduction. So for my research, I performed an analysis on the efficacies and chemical composition of 4 different COVID-19 vaccines. The first of which is the Pfizer BioNTech vaccine, which is an mRNA vaccine. Another mainstream vaccine is Moderna, which is also an mRNA vaccine. And the last 2, Covishield and Covaxin, are based primarily in India. Covishield is an Indian manufactured version of the Oxford AstraZeneca vaccine. Covishield is a chimpanzee adenovirus vaccine, and it uses an adenovirus to inject the mRNA to create those spike proteins to combat the coronavirus. And Covaxin is a more traditional vaccine using a dead coronavirus. So the main issues throughout the time that COVID-19 has persisted and throughout the pandemic are mainly common in the gaps in the demonstration of clinical efficacy and the hurdles that we have to jump over to suffice global demand for the vaccine. And in general, gaps in knowledge for the therapeutics, transmissions, and social sciences pertaining to COVID-19 remain largely uncovered. To better understand this, I wanted to compare these 4 vaccines, which range from 4 of the largest vaccine categories, in order to better understand our current situation. And the question I developed is, how do each of these vaccines compare to one another, and which projects stronger traits in protecting the population? So this is just a short literature review. I found on the net many existing scientific papers discussing in depth the components of the COVID-19 vaccine and its efficacy percentages. And that was incredibly helpful to my own research. And the origins for my own research for this comparative study comes from the fact that there's not much research on specifically comparing certain types of vaccines altogether. So I went ahead and compiled my own review of various vaccine types, including Pfizer, Moderna, Covishield, and Covaxin. So this is my methodology and results. The main way I acquired my data was through scholarly articles and research databases, including PubMed and NCBI databases. And I also took statistical findings from company fact sheets. So each of these companies, Pfizer, Moderna, etc., they all have safety data sheets or fact sheets containing the exact biochemical names for their chemical ingredients and other types of statistics that are useful in taking apart and dissecting each of their vaccines. So in terms of efficacy, I found that Pfizer had a 95% efficacy, Moderna had a 94%, Covishield had a 91%, and Covaxin had a 78%. And if you look to the right, I constructed my own table just organizing all of the statistics. And in general, I found that the vaccines can be generalized to a series of lipids, salts, and sugars. Each of these vaccines have their own active ingredients, which is what sets them apart. And to conclude, I just wanted to reiterate some of my results. And my main question was, how do some of our most popular COVID vaccines compare to one another? And to answer that question, through extensive research and analytical thinking, I found that I created a review displaying which vaccines had the highest efficacy and why that may have been on a more pharmacological level. Thank you for listening.
Yeah, thank you very much. Very good. Next up, we have Aditya Srivastava. I'm sorry, did you put your topic in? I'm sorry. Aditya Srivastava, please go ahead.
Okay, Michelle.
Yeah, please go ahead.
Sharing screen, PowerPoint, share. Can you guys see my screen?
Yes, go to the first page and then do present. Yes.
Hello, my name is Aditya Srivastava. I just recently, a week ago, started 9th grade, and I go to Osceola High School in St. Petersburg, Florida. My topic and my introduction was the neutralization of the COVID-19 vaccine, or SARS-CoV-2. How does it work? And some side questions I picked up along the way of my research. How does the COVID vaccine work? What is the efficiency rate? Is it why take the vaccine? And is it safe? So I studied the efficiency of the COVID vaccine, and the efficiency for Pfizer is 95%. Scientists from the CDC, FDA, Yale, big branded names all have said that the COVID vaccine is very safe. There is a near 99 to 100% chance that you will not get hospitalized once you get the vaccine or your vaccine. Side effects. You will have some side effects within the first 2 weeks of the vaccine. Displaying your immune system fighting the vaccine, or a weakened version of COVID. Some symptoms for COVID-19 are cough, fever, headache, fatigue, and difficulty breathing. The way the COVID vaccine was developed was the scientists wrapped molecules of mRNA with lipids. This was because our cells are lipophilic, which basically means lipid loving. By definition, that means if they're lipid filled, that means they'll be able to go into our immune system easily. That creates viral spike proteins and activates our immune system, creating T cells to combat our virus. One thing I got along my research was, can you still spread the virus if you're vaccinated or vaccinated, and if you should still wear your mask? While you may have a significantly lower chance of spreading the vaccine when you're vaccinated, I would still recommend that you still wear your mask, because there's always a chance to still spread the virus. So still keep wearing your mask, still keep using hand sanitizer, do whatever precautions are necessary. In order to reach herd immunity, or when the vaccine is the most efficient, is after 2 weeks after the vaccine is when you can reach herd immunity. As of right now, there has been no long-term side effects of the COVID vaccine. Short-term, within the first 2 weeks, are just bruising of the arm, and you'll feel like you have a cold, but other than that, there should be no more side effects. I'd recommend you see a doctor if they are. My summary conclusions was restating the question: how does the COVID vaccine work? Is it safe? Do I still wear a mask and social distance? Answer to all of those is yes. And any questions?
Great. Great job, Aditya. Wonderful. Next up, we have Om Patu. He's a junior in Cumberland Valley School. His topic is the effect of stress on insulin production across different generations. Go ahead, Om.
All right, so yeah, that's me. I'm Om. And my topic is the effect of stress on insulin production across different generations. So the main question that I wanted to answer through my research was, is there a connection between insulin production, stress, and age? And the goal of the research was to establish a clear connection between insulin production, stress, and age, so further research can be done on this topic. I have established a clear connection, as you guys later find. And the reason why I wanted to do this topic was because we all know that stress is not a very good thing, and it's not very good to be stressed. It has a lot of bad effects on your body, negative effects. And a lot of people also have diabetes in this world. And I just wanted to see if there was a connection between stress and diabetes so that maybe anti-stress treatment can be introduced for diabetics. So here's a literature review. This isn't really literature, I guess, but it's just some new technology that I found was pretty cool. At least the first one, Semglee. Semglee is a new, cheaper brand of insulin that just got approved for the FDA. As we all know, insulin is one of the most overpriced medications in the market right now. It's basically monopolized because the people that buy it can't afford it, and Semglee aims to combat that. Another research paper that I found was really interesting and if anybody has an interest in stress and its effects on hormones in general, I would read this paper because it focuses and delivers this message very well. As you can see, stress causes many imbalances in hormones, and that could be through production and the hormone itself, the effect of the hormone. So here's my methodology and results. So my analysis mainly consisted of graphs and statistics. This is one of the graphs right here. And the graph over here basically just shows the percentage of the population with diabetes and how it increases as people get older. And another thing that I found was stress and glycemic control share a direct relationship. I found this through another graph and a set of information, which I didn't include in here because it didn't fit. But it's basically what it says over there: more control, more glycemic control, more positive stress, and less glycemic control, less positive stress. Here are my most significant findings. In most cases, stress decreases insulin production and insulin effectiveness because of the inhibiting properties regarding hormone production. And emotional stresses effect on insulin production decreases with age due to the older generations being more suited for emotional stress. They have more experience with life and how to handle it, so they can experience and endure more of it without having negative effects. But the physical stress that comes with age, just growing older, you go slower, your back starts hurting, there's more damage in your body, and that causes a drastic decline in production. So here's my conclusion. As the same question, does a connection between insulin production, stress, and age exist? I clearly made a connection there, and I hope that this connection can become a building block for further deeper research regarding insulin and stress. And I'm personally probably going to do more research on this topic just to build off of this. And final thing I want to talk about are limitations regarding my research. And the only limitation that I found were just a lack of statistics regarding age and insulin production, like a direct relationship between those two. But I found a few, as you can see with the graphs. So it ended up pretty well. Thank you for listening to my presentation.
Yeah, very good, Om. I hope you continue with your research. Next up, we have Vandana Vijay Kumar. She's from Forsyth, Georgia. Her topic is alternative motor strategies effect in hand selective areas of the brain in prosthetics.
So hi, my name is Vandana, and I did my research on prosthetics, mainly hand amputees, and how it connects to certain hand selective areas of the brain compared to non-amputees. Hand hand-selective areas are a key component of this research, and they're basically the regions of the brain which are responsible for controlling the hand and its other related areas. So my paper's topic was both talking about the alternate movement or motor strategies of amputees compared to regular people, and that these strategies are able to pave the way for future innovations and technologies such as neurologically controlled artificial limbs. The artificial limbs that are in production now are something that needs to be manually fixed, so neurologically ones would be more efficient. Since there are not a lot of current studies available, I used experiment conducted by the paper Artificial Limb Representation in Amputees in order to provide a basis and compound all of the most reliable and current studies into this paper. Experiments like these and the analysis that I provided can help set the foundation for more advanced artificial limbs and to help aid amputees regarding the motor movement in their everyday life. One area of weakness is that the idea behind these studies and the effect of them is very important and powerful. However, there needs to be a significant amount of additional research and more funding, as when these artificial limbs are sold to the public, tens of thousands of dollars will be needed for access for each one, and for any type of surgery that's involved with connecting the nerves of the hand-selective areas to the phantom limb. So I first studied background information on what the two main areas of the brain responsible for hand motor movement are, and those are the sensory motor cortex and the visual cortex. The experiment goes on to emphasize the importance of movement and visual activity in the nervous system. After the experiment took place with an MRI scan, a questionnaire, and a series of behavioral tests to see all the relationship between the motor and visual, there was strong evidence of increased activity in these areas as communication. So basically a threefold coupling. This shows that prosthesis users are able to adapt very quickly to maximize other organs and nerves in spite of the amputation, as shown by the threefold coupling. As said by the previous slide, the experiment provided a solid basis upon which verified evidence of the alternate motor strategies were identified, since different regions of the brain, motor and visual, were highlighted strongly in some areas and overall just increased in the highlighted areas during the MRI scan. Because of this, bionic limbs are a definite possibility, and there are already some undergoing clinical trials. With the advanced artificial limbs, amputees will be able to control it just like their regular limb, and they'll also not have to deal with the burden of a lifeless prosthetic. They will be able to live their lives in the same way as non-amputees, and if this technology develops further and there's enough funding to host these clinical trials, people will be able to buy these limbs and also create a new generation of artificial limbs. And that's it. Thank you for listening.
Thank you, Vandana. That's a very good topic, prosthetics. It's a very unique topic indeed. Okay, so next up, we have Pranav Varshney. He's from Missouri. His topic is unique implications of creatine therapy and exercise as a means to mitigate and treat depressive symptoms and depression along with other mental health issues. So go ahead.
So as Kojo said, I'm Pranav Varshney, and the premise of my research is based off of unique treatments such as creatine therapy and exercise, and how it treats depression and it mitigates depressive symptoms, along with other mental health issues. So first of all, we have to give some insight towards what depression actually is. And depression is a mood disorder, and quite frankly, it's the largest mood disorder that affects the world. There are, as of 2012, there are a total of 350 million people who suffer from depression, and those numbers are only rising. So in the following research proposal, we'll be discussing the deeper impact of the crucial crisis on the anatomical brain. And more importantly, we're going to discuss different types of treatments that will help in mitigating depression. So for a table of contents, we will first start off with our literature review, which is current relevant research, as well as we're going to move on to methodology, so how our data was collected, and some of the results we were concluded. results analysis, which was the most significant findings, and finally the conclusion, which was limitations in my contributions. So there were really mainly like 3 or 4 different articles that I saw online, and most of them were talking about creatine for the treatment of depression, of course. And basically what it was talking more about was the fact that creatine develops phytocreatine, or a chemical that is actually more stable than ATP, or the standard cellulose of energy. So what we're talking about is employing creatine therapy, or like dosage of creatine, and kind of implementing that into more of like a antidepressant drug type therapy, and overtaking the lack of energy that the brain gets. Because in depression, what happens is the brain kind of shuts down and there's not enough energy that's pumped to it. So through creatine and its more stable energy source than ATP, or the general energy source, we'll be able to pump enough energy and kind of get the brain out of its depressive state. So that was the main focus of creatine. Now for exercise, we talk more about the effects of exercise in the prefrontal cortex, the hippocampus, and the corpus callosum, and how its structural neuromarkers are targets for exercise-based treatments for depression. Next, for the methodology, there were 5 kind of steps. The first was hypothesis building, and this was mainly like personal orientated. So the reason I kind of chose to talk about depression and other mental health issues is that it has affected myself and a lot of my classmates and friends a lot. So I wanted to be able to reach out to the world and kind of introduce some new aspects or new possible treatments that doctors aren't really looking at a lot. Next step was the research exploration, which was more of PubMed and NCBI and ResearchGates, kind of searching up for different types of formal research papers that were written on that, and kind of, looking at some quantitative data, crunching those through my own numbers, my own formulas, and presenting a experimentation method, which finally results in, which came up to the results and conclusion that I will show you. Results analysis. So these are the 3 main numerical results that I came up with. This bottom table is based off of the different diets. So you can see omnivores, pesco-vegetarian, lacto-vegetarian, and vegans. And at the bottom, you can see the depressive symptoms. And the brackets and the CESD score talks about depressant symptoms, and you can see that the bracketed number, so 16.19, 25.48, 28.11, and 28.40, they increase as you go towards a more plant-based diet. So that's pretty important because it's drawing a direct link between vegetarianism and an increased amount of depression. And that makes sense because you're not getting enough creatine or creatine supplements through plant-based diets. And I'll talk more about some of the limitations that came with this individual study that I did, but more talking about depression, more talking about exercise. We have the meta-analysis of exercise on depression. So the standard mean difference was around 0.66, 0.65 for benefiting of exercise in lowering depressive symptoms. Next is the lasting benefits. And as you can see, the standard of error from the graph is much larger. And kind of a reason for that is there's not enough online material available to come to a conclusive result for it. So finally, in my conclusion, creatine and exercise are decisive in treating depression. And I truly believe that both of them individually have the possibility of overrunning antidepressant drugs. Creatine and exercise are a very new topic, and one of the limitations that I saw was that there is no rigorous method of research. My proposal, and I think this is my main contribution, is to run a tri-pronged research scale, one that uses regular antidepressant drugs through a set n equals, let's say 50, one that uses placebo, and one that uses creatine drug therapy. So through this tri-prong research, we will be able to see what the true effects of creatine is compared to placebos and compared to conventional antidepressant drugs. So that's one thing I really noticed, that there's really not that much research. I think a total of only 4 research papers came up when I typed in creatine. And creatine obviously is more of a pre-workout type of drug. So most of the research papers were leaning towards workout rather than treating depression. And exercise itself talks more about different ways to alleviate more addiction type of treatments. For example, addiction to alcohol and addiction to drugs. But it doesn't talk about depression and mental health issues. So I feel like creatine and exercise can really open the gates to doctors for finding new and I think more beneficial treatment possibilities. So thank you, and if you have any questions, I have my email right there, phone number right there, and thank you to Coach Joe and Dr. Raj for supervising me. And then credits to Slides Go, which was what I used to make my
great job, guys. I apologize if it ran over. I didn't anticipate it, but I'm not going to stop my students from presenting or expressing themselves. This is their work. I'm sorry if it's running later than expected, but I'm taking so much pride in all the hard work that my students have put in. So I'm recording this, and let's go ahead with Avinash. Avinash is a junior in Great Valley, Pennsylvania. His topic is comparison of different COVID vaccines in terms of demographics, mechanics, side effects, and chemical compositions. Go ahead.
Oh, can you see that?
Yes, absolutely.
All right, sounds good. Okay, my name is Avinash Kunde. I go to Great Valley High School, and I'm in Pennsylvania. All right, so let's continue. So my introduction. So COVID actually needs no introduction. In the United States, there's been 624,000 deaths so far, and worldwide there's been about 4.38 million deaths so far. So it's had serious implications. And obviously my topic area is the coronavirus vaccine. And some questions I went into the research thinking about are what are the differences between the vaccines, specifically looking at the ones that are available in the US right now. So the Moderna, the Pfizer, and the J&J vaccine. So which vaccines are the most effective and for what reason? And then what are some mechanisms through which the different vaccines work through? And my objectives would be to give people a better understanding of COVID vaccines so they can make a more educated choice on which one to get. All right. So this is my literature review. So primarily for this research, I'll use the fact sheets, the COVID fact sheets and the safety fact sheets that are actually released and reviewed by the FDA. And since I was mostly comparing the US vaccines, I have the fact sheets. I use the fact sheets for the Pfizer vaccine, the Moderna vaccine, and the J&J vaccine. So I also use some previous scientific literature on mRNA vaccines because they aren't new. They're like there have been mRNA vaccines for the Zika virus before. So I use that to further my understanding on mRNA vaccines. All right. Okay, so here, obviously, I think that graphs are important. They kind of show you a better picture of what's happening. So here are my results. So if you look at the left graph, this is just showing you the efficacy comparison between the three US available vaccines, J&J, Moderna, and Pfizer. And you can see that Pfizer and Moderna, they're pretty similar in their efficacy in that there's no statistical significance between their efficacy. But when you look at J&J, it's much lower than Moderna and Pfizer. And the reason for this is that Moderna and Pfizer have 2 doses compared to J&J's 1 dose. And I'll kind of explain a bit more about that. But moving on a bit. So why do why do Moderna and Pfizer cause more side effects than J&J? And the reason for that is that obviously 2 doses, and essentially there are these proteins called cytokines, and these proteins command growth of immune cells. So the first time you get a vaccine, cytokines are released and you have an immune response. And the second time, if you take a Moderna or Pfizer shot, more cytokines are released, which result in a stronger immune response. And that's why, if you take a Moderna and Pfizer shot the second time, you obviously have more side effects and a bit more a stronger side effect. And you can see in this graph, this middle graph, a higher percent of the population had headaches on their second dose of Moderna and Pfizer. Now, obviously, J&J doesn't have a second dose, so I can't really have a chart there for that. So that's the reason why J&J has a lower efficacy. It doesn't have the cytokines. There's not that many in the first shot compared to the second shot. And this is the third graph, all the way on the right. This is just showing you the age group comparison. So if you look at it, the gray bar is essentially the older age group, and the blue bar is the younger age group. And you can see that for the J&J, the older age group experienced a higher efficacy than the younger age group. For the Moderna shot, the older age group showed a lower efficacy than the younger age group. And for the Pfizer shot, they were roughly comparable. So this is like some of my most significant findings I found through my research. All right, so conclusion. To conclude, all the COVID vaccines have their individual pros and cons, and these pros and cons need to be weighed for everyone to make an educated guess or educated choice on which vaccine is best for them and their own situation. But what can be said with a certainty is that getting the COVID vaccine for the majority of the population is better and safer than not. So I would implore people to definitely get back.
Thank you very much. Beautiful presentation. Next, we have Sneha. I'd like to award her as the most hardworking kid ever. She's always there like a busy beaver, like always asking questions, how to get done. So I'm sorry I kept you till the end. I apologize, but here you go, Sneha. Her topic is connection between COVID-19 variants, Gamma and Delta variant, and demographics using...
Hi everyone, I'm Sneha, and today I'm going to be talking about that long topic that's on my slide. I'm not going to say it, okay? So my research paper revolves around topics like COVID-19 pandemic, the demographics, and most important, COVID-19 variants. The specific question that my research paper is set to answer is basically about whether there is a relation or a connection between COVID-19 variants and demographics. So literature review. The big difference between my research paper and other research papers is that other research papers generally talk about COVID-19 and not like specifically about each prominent variant. So that's what my research paper kind of does. So the methodology, it's basically on the slide, but I got a data set from a credible website, and I was able to download that as a CSV file. And then I used Python to extract that data, and then I analyzed it, and I manually made pie charts using the SmartArt tool on Word. And then I developed theories, and the results are kind of dubious, but you'll see why. It's due to some limitations. So the conclusion, my research paper attempts to answer the question that I previously stated. But however, due to the limitations such as insufficient data and evidence, the theories that I've developed in my study can be invalidated by other pieces of information. So this research paper mainly acts as a stepping stone to inspire passionate researchers all over the world to solve this global problem. Thank you. Thank you for your time and patience.
Bravo, Sneha. Very, very good. Excellent. Next up, we have Shwant. Shwant, I don't have your topic, unfortunately. So Shwant, can you please go ahead?
I did my research on machine learning. So the multitude of functions that machine learning holds has given it an important role during the pandemic. The term machine learning can be defined as an aspect of artificial intelligence capable of creating systems of data and being able to learn from the data and make predictions. The most popular algorithms used that are under machine learning is naive Bayes, KNN, random forest, and decision tree. So while researching machine learning, I wanted to find out how it has helped during the pandemic, and in doing so, I had to research the algorithms I stated right now and how these algorithms have solved various problems.
Sorry, Shwan, did you forget to change? Okay, yeah, go ahead. Sorry.
So current research on these four algorithms these four algorithms are most optimal for solving most problems, such as vaccine hesitancy, patient prediction analysis, recovery, and much more. So for the results, something interesting that I found is so various algorithms are more efficient in various problems. So for example, KNN is best used for analyzing COVID patients. And within KNN, there are various types, and I found that an enhanced KNN does significantly better than normal KNN. And then on the right, I've organized various data sets, comparing not just enhanced and normal, but other types. So to answer my question, how has machine learning helped during the pandemic? Machine learning has generated highly accurate solutions to multiple problems in varying sectors. Naive Bayes was a classifier that was depended on for COVID-19 detection. Random forest used decision trees for creating outcomes with vaccine data sets. KNN utilized regression analytics to perform prediction analysis on COVID-19 patients. Decision tree applied data mining algorithms to project survivability rates depending on age, gender, and various other factors. Some of the contributions I've made during my time researching, I now analyze applications of machine learning for various problems. I also created data tables comparing the performance metrics of several types of KNN.
Very good, Shwan. Very good. I mean, thanks for introducing us to so many concepts of machine learning. Thank you so much. Next up, we have Ashwin. Ashwin is a rising senior in Bridgewater. His topic is the potential of biosensor technology application in COVID-19 detection. Go ahead, Ashwin.
All right, as Coach Joe said, my name is Ashwin Ajit, and I'm going to be in 12th grade, and I go to Bridgewater Raritan High School. My research topic is the potential of biosensor technology application in COVID-19 detection. So my topic is the possibility of alternative methods of COVID-19 detection using this biosensor technology. So what are biosensors? Biosensors are made up of two basic parts, a bioreceptor and a transducer. The bioreceptor is any biological element that can give off a response when it detects a molecular interaction or analyte. The transducer will then take the signal and it will send it to a device so the data can be monitored. The research questions I wanted to answer were, what are the current methods of diagnosis used and what are the areas for improvement? What exactly are biosensors? What are the biosensor alternatives that could be used? How do they work and how effective are they? My objective with this research was to research these questions and be able to draw my own conclusion from the data I got. Literature review. Lots of research has been done on methods of detection that utilize biosensor technology. The commonly used RT-PCR works by reverse transcriptase and DNA polymerase that reveals the presence of SARS-CoV-2 through a fluorescent signal. The four main methods of biosensor detection that I researched were SPR, which is surface plasmonic resonance, PHET-based biosensing platforms, electrochemical biosensors, and SARS biosensing. These are newly developed biosensors that will detect COVID-19. SPR works by monitoring the body's antibody levels to see if the body is immunized against COVID-19. PHET-based biosensing platforms are able to pick up small amounts of analyte instantaneously. They are conjugated with specific antibodies against SARS spike protein. Electrochemical, also known as EC biosensors, are known for their simplicity. They can make use of fluorine-doped tin, oxide electrodes, and screen-printed carbon electrodes immobilized with COVID-19 antibody to detect. SARS biosensing is an alternative to the commonly used colloidal gold signal reporting. Methodology and results. So, the research on these alternative methods show many advantages to further developing biosensor diagnostic methods. Biosensors can avoid the lengthy and difficult process of labeling and instead focus on monitoring subjects in real time. Many of these methods are highly sensitive, such as the graphene-based PHET biosensing, and the SARS biosensors. Biosensors can also be affordable, easily miniaturized, and mass-produced, such as EC biosensors. These attributes also make them useful for on-site diagnostics. As you can see in this table, the commonly used RT-PCR takes around 4 hours to complete and only has a sensitivity of 71%. This means that when you take an RT-PCR test, you'll normally get the results the day after. And within this time period, you could spread COVID-19, if you have it, to many people. But if you look further down the table, you see plasmonic biosensors and different types of biosensors that only take a couple of minutes to give a result. These are also with higher sensitivity, which means that they can detect earlier on with low concentrations of the viral DNA. So my conclusion, biosensors are a promising and rapidly growing field that can aid with COVID-19 detection and is a technology that should be further developed to be used with future viruses. Many biosensing methods have high specificity and sensitivity, all while taking much less time to deliver a result than current methods such as RT-PCR. However, RT-PCR is a reliable method that has been thoroughly tested, unlike newly developed biosensor forms of detection. More research will be done to give credibility and increase specificity. I was interested in this topic because recently I went on a trip to India, and to make sure that we hadn't gotten COVID-19 on the plane ride, we all took the rapid antigen COVID test, and my sister got a false positive with her test. So I want to see how exactly these methods of detection work behind the scenes. Thank you for listening.
Very good, Ashwin. Awesome. Is Tristan here? He was the last person. Other than that, we've finished. I just want to thank Dr. Raj. He's not here. I mean, it wouldn't have been possible without him and his expert guidance. I still remember the first day when all the students were there and we were discussing topics. And we've come a long way. I'm so proud of each and every one of you, the hard work that you've put in. And there's no more torture emails from Coach Joe threatening and everything. And I'm really going to miss you guys. And I just want to wish you the best. This is just the beginning of your research. I want you guys to contribute, to continue with your passion. Each one of you have done an amazing job. And thank you, parents, for trusting in me with your children. And they are all amazing. They've done wonderful job with it. So sorry for keeping you guys for such a long time. I'll let you all go. Enjoy the rest of your summer, and seniors with your college applications and everything, and stay in touch. Bye now.
Thank you, Joe.
Thank you, Coach Joe.
Thank you so much, Coach Joe.
Thank you, Coach Joe.
Thank you. Good night.
Thank you so much.
Bye. Thank you so much. Good night.
Guys.
Yeah. Also, thank you for everything, Coach Joe.
Yeah.
I really appreciate it.
Thank you so much. Every one of you, you did an amazing job. Yeah.
Thank you.
Quick comment.
Yes, Anish.
So I just wanted to say that you and Dr. Raj have helped me a lot with how to make a research paper and how to design one. Because initially when I started, I was pretty hesitant on what topics I should be choosing, how to format it correctly. And so I just want to say thank you for all you've done, because you've really helped me on how to make research papers, how to format it, and how to present it in a clear audience. So I just like to say thank you a lot for helping me.
Yeah. Thank you, guys. I mean, it was amazing working with each one of you, right? I mean, I pushed you guys so hard. I mean, I was constantly pounding you guys week after week, but you never complained. You all did wonderful. You're all amazing. I had such a great time with it. I learned so much from you guys. Bye now. Bye.
Thank you so much, Coach Joe. Bye. Have a good day.